Successful and unsuccessful persons do not vary greatly in their abilities. They vary in their desire to reach their potentials. Hello, good morning students. This is Papiya Miss and you are with the online class of Ananda Multimedia School and College. Welcome back to the biology class. Today we are on the chapter 11, Reproduction in Organism. My previous class was about the Microsporogenesis. In today's class, I will discuss about the Microsporogenesis. What do you mean by Microsporogenesis? Microspore means the male gametes and genesis means development. So, the development of male gametes are known as the Microsporogenesis. Before going to the main topic, we should know something details about the structure of a typical flower. As usual, this is a typical flower. This one is the peduncle. This is the thalamus. This one is the sepal, petal, stamen and carpel or pistil. Today, our center of attention will be on the male sex organ. That means the stamen. This is the stamen and pollen grains are produced inside the anther. This one is the structure of a typical stamen. This one is anther and this long thread-like structure is known as the filament. The filament attaches the whole structure to the flower. This one is anther, this one is filament. Okay students, and this is the TS of the anther. If we make a transverse section, we will get a structure like this. 4 chambers. These are the pollen grains and these are the pollen sacs. And this is the anther, this is the filament. And the TS will be like this. 4 chamber structures. And pollen grains will produce here. I will describe the whole structure after showing the figures. These are the pollen grains. Pollen grains look like this. This is also pollen grain. This is also pollen grain. Now I will show how the pollen grains are produced in the or inside the Microsporangium, right? Students, I have divided my lecture in 4 segments. Number 1, TS of the anther. Number 2, development of pollen grains in the Microsporangium. Number 3, structure of pollen grain. And number 4, development of male gametes or Microsporogenesis. 1, 2, 3 and 4. Our main focus will be on the number 4. But we should know something more, something detailed about these points, because there are some curious students who wants to know detail about anything. These points, that means 1, 2 and 3, will help you to know more detail about the Microsporogenesis. So we should know these structures. These 1, 2 and 3 will not will not sit in your exam, but you should know to understand better the development of Microsporogenesis. You should know this, okay? First one is the excuse me, TS of anther. Okay, I am starting. Suppose this is an anther. This is an anther. This one is the anther and this one is the filament. Okay, So the total structure is known as the stamen. It has two lobes, 1 and 2, and in each lobe there are 2 cavities. If I make a TS through the anther, what will I see? I am making the transverse section of the anther. Then I will get this type of structure. And in each lobe there are 2 theca. 1 theca, 2 theca, 3 theca and 4 theca. In each lobe 2 theca, so it is dithecous, and in total there are 4 thecas. 1, 2, 3 and 4. So an anther has 2 lobes and it is dithecous. What happens inside the anther? There are 4 sac-like structures. These are known as the Microsporangium. Gium or gia means the sac or the container. So this is Microsporangium. It has numerous sporogenous cells or sporogenous tissue. Sporogenous cells or sporogenous tissue, and it is covered by a layer. It is known as tapetum. It is known as tapetum. What is the function of this tapetum? It provides nourishment to the developing sporogenous tissues, because these sporogenous tissues will give rise to the microspores, and after that these microspores will give rise to the pollen grains. So these tissues are very important and they get nourishment from the tapetum, and we will find 4 types of layer inside the theca. That means E, M and T. E means epidermis. The outer covering is known as the epidermis. These are epidermal cells. And next, endothecium. This is endothecium. What are they? Endothecium. That means epidermis, endothecium, and this is the middle layer. And after that, T means tapetum. So, epidermis, endothecium, middle layer and tapetum. Tapetum provides nourishment, and these 3 layers help in the splitting. That means dehiscence. Okay. Now I will draw what happens inside the Microsporangium. It is also Microsporangium, Microsporangium, Microsporangium and also Microsporangium. What happens inside the Microsporangium? Excuse me. Suppose this one was our Microsporangium. And if we take only one cell inside from the Microsporangium, it will be like this. These are the sporogenous cells or sporogenous tissue. This sporogenous tissue will behave like PMC. That means pollen mother cell. Pollen mother cell. Rather, you can say MMC. Here MMC means microspore mother cell. Both one is correct. I have taken only one cell from this structure. This is the PMC or MMC. That means pollen mother cell or microspore mother cell. And it will undergo meiosis. This is 2n in number, that is diploid. Meiosis. Meiosis. Do you know what will happen in meiosis? I know you know. From one cell we are getting 4 cells. So 4 cells, but this 4 cell will not like this. it will be tetrad, and its pattern of arrangement will like 1, 2, 3 and 4. We will call them as haploid nucleus in. After that, I am drawing here what happens. This is haploid nucleus, and its structure after some time will be like this. No, I am showing accurately. 1, 2, 3, and where is another one? Another one will be on the back side. So it is known as the microsporogenesis tetrad. Tetrad means 4. 1, 2, 3 and another one is in the back side. So microsporogenesis tetrad remains attached closely. After some time, separation takes place. Separation, and after separation it will give rise 4 pollen grains. So we can say that from 1 PMC or from 1 microspore mother cell, we are getting 4 pollen grains. So I have finished TS of anther. It was TS of anther and development of pollen grain in the Microsporangium. Clear, students? Now, structure of the pollen grain side by side. This one is the pollen grain. What will be its structure? Structure of pollen grain. This is a typical pollen grain. It has, this is the plasma membrane. After that, this one is the inner layer, and after that the ornamented outer layer. Students, this is a typical pollen grain. It has 2 layers. Outer one is known as the exine. Inner one is known as the intine. This one is the vacuole. This one is the plasma membrane. And nucleus. Cytoplasm. Now, another important structure. That means in the outer layer or exine, there are some pores throughout the exine. These pores are known as germ pore. In dicots, you will find 3 germ pores. That means in the place there is no exine. Only intine and plasma membrane. Absence of exine. And I want to say something about exine and intine. The exine is hard. What's hard or tough? Thick, and it is ornamented and also resistant. It is made up of sporopollenin, the most hardest organic substance in the world. No acid, no enzyme can degrade this. That means the exine. It is the toughest or hardest organic substance in the world. No enzyme can degrade it. This one is the exine. Now intine, this structure, this. Intine is thin, delicate and elastic in nature. This one is intine. And you know vacuole. This one is the vacuole, plasma membrane, nucleus and germ pore. Now mitosis will take place inside the pollen grain. I am just erasing this structure and I will draw how the mitosis is taking place inside the pollen grain. Suppose this one is our pollen grain, this one is the plasma membrane, this one is the intine and this one is the exine. What happens? I have drawn the germ pores here, 1, 2 and 3 germ pores and nucleus, vacuole. First, mitosis. According to the law of mitosis, you know that from one cell we get 2 cells. First of all, karyokinesis will take place. That means the nucleus will divide first. And what will we get? What is this? Now I should draw with black lines and this should draw with blue lines. That means it is intine. Oh shit. And this one is exine. Right? Karyokinesis has taken place. So from one cell we are getting 2 cell structure. This one is the generative cell. Generative cell. And this whole structure is vegetative cell. Vegetative cell and exine, intine as usual, TINE, plasma membrane, cytoplasm, and now this one is pollen grain. This is also pollen grain. This is generative cell and the whole structure is vegetative cell. The generative cell remains attached with the plasma membrane at first. After some times, what happens, students? I am erasing the labeling because so that I can show you the next structure. What happens then? This one is the plasma membrane, intine and exine. You know, students, the pollen grain performs mitosis twice. Number 1, before pollination in the anther, and number 2, after pollination on the stigma. Sooner or later, the generative cell will come close to where the germ pore, and it will take a spindle shaped structure. spindle shaped structure, and this is the vegetative cell. The vegetative cell is larger in size and generative cell is smaller in size. This is two cell structure. Generative cell has larger nucleus. Vegetative cell has smaller nucleus. It has larger cytoplasm. That means the vegetative cell has larger cytoplasm, but the generative cell has smaller cytoplasm. Again, the vegetative cell contain different types of cytoplasmic organelles, reserve foods and cytoplasmic RNA. Which one? Vegetative cell. But generative cell, in generative cell you will not find any type of these materials. Again, the vegetative cell is also known as tube cell, and the generative cell is known as the gamete cell, because from the generative cell gametes will produce, and from the vegetative cell pollen tube will produce. And this structure, these three structure will happen before pollination. Before pollination, most important, pollination in the anther. In the anther. Now I will show after pollination what happens, students. Should I rub this? I am rubbing this. Oh, I didn't draw the cytoplasm. Before pollination in the anther. Now I will show what happens after pollination on the stigma. The pollen grains fly and sit on the stigma. And what happens? This pollen grain, this one was like, and plasma membrane, intine and exine. Suppose our jumper is here, and you know the outer exine is the hard or tough, resistant, ornamented layer. So God has gifted something to the pollen grains. That means in some places on the exine there are some weak points, and these are known as the jumper. And in this case there is no sporopollenin, and it is like this structure. The structure should be blue. There is no sporopollenin. What happens? In this case, the cytoplasm with vegetative cell and generative cell come through the jumper. Why? What is the logic behind this? Can you say? The pollen grains fly and sit on the stigma. The pollen grains have some receptors, and also the stigma has some receptor. If the receptor of pollen grains match with the receptor of the stigma, then the stigma will allow the pollen grain to germinate, otherwise not. Clear? If the receptor of the pollen grains match with the receptor of the stigma, then the stigma will allow the pollen grain to germinate, otherwise not. The pollen grains will destroy. And if the receptors are matched, then what happens? Receptors are matched, right? Then the stigma becomes so pleased and it feeds sugary fluid to the pollen grains. It feeds sugary fluid to the pollen grains, and as much as the sugary fluid the pollen grain will take, it will become larger and it will feel some pressure inside it because it has taken much more sugary fluid from the stigma. It feels some pressure inside this. So the cytoplasmic membrane and generative cell and the vegetative cell comes through this point, this aperture, and makes a long hollow structure which is known as the pollen tube. Students, you know that the pollen tubes of the nucleus of the pollen tube, the nucleus of the generative cell is much more active than the nucleus of the vegetative cell. It should be active because it has to produce two gametes from it. Now what will come? This one is suppose the nucleus of the vegetative cell, and suppose this one is the nucleus of the generative cell and cytoplasm. Okay, clear? In the next stage, what happens? I didn't level because you know the leveling. What happens here? Suppose this one was our not so long now, and blue, it was the intine and exine, two jumpers. What did I say? I told that suppose this one is the nucleus of generative cell, okay, and nucleus of vegetative cell. Nucleus of vegetative cell. What happens now? This one. That means the nucleus of the generative cell will undergo second mitosis. Second mitosis. I am drawing the structure here for better understanding. Right, students? After second mitosis, it will produce two male gametes, and before that, callus plug. What is callus plug? This is a plug. It is made. Why it is made? So that the cytoplasm and also the generative nucleus and the vegetative nucleus cannot flow back. So it prevents the backflow of these materials. The callus plug is made to prevent the backflow of the material. Now, second mitosis will take place. Mitosis will take place, and from the law of mitosis, you know what? From one cell we are getting two cell structure. Oh, what is this? I am drawing exine. Callus plug, nucleus of generative cell and nucleus of vegetative cell. Now from one male gamete we will get two male gametes. because second mitosis has taken place here. So the entire structure, that means the two male gametes, the nucleus of the vegetative cell and also the pollen tube together known as the male gametophyte, together known as the male gametophyte or microsporogenesis. Students, we have finished our lecture. If you have any problem understanding this lecture, watch the video again and again, and I think that your problem will be solved. Okay students, till today, take care, keep watching, see you later.